There's something inside you right now, something that wasn't there a century ago, something that's accumulating in your organs, coursing through your bloodstream and embedding itself in the most intimate tissues of your body. It's not alive, yet it's everywhere. It's not a virus or bacteria, yet it may be reshaping human health in ways we're only beginning to understand. These are microplastics, tiny fragments of synthetic polymers that have infiltrated every corner of our planet and, most disturbingly, every corner of our bodies. They're in your brain, they're in your heart, they're in your lungs, your liver, your kidneys, and your reproductive organs. If you're pregnant, they're crossing the placental barrier into your developing child. If you're a new parent, they're in your breast milk. From the moment we're born until the day we die, we're accumulating plastic inside our bodies, particle by particle, meal by meal, breath by breath. The story of how we discovered this invasion reads almost like science fiction. For decades, researchers studied plastic pollution in the oceans, documenting how sea turtles mistook plastic bags for jellyfish, how seabirds fed plastic fragments to their chicks, how microplastics accumulated in the tissues of fish and shellfish. But it was 2022 when everything changed. That year, scientists made a discovery that sent shockwaves through the medical community. They found microplastics in human blood. This wasn't a small, isolated finding. The researchers examined blood samples from 22 healthy volunteers, and they found plastic particles in 17 of them. That's 77%. These weren't people who worked in plastics manufacturing or had unusual exposures. These were ordinary people living ordinary lives. And inside their veins, flowing through their circulatory systems, were tiny fragments of polyethylene, polystyrene, and polymethyl methacrylate. The plastics we use to make grocery bags, coffee cup lids, and plexiglass were circulating in human blood. But blood was just the beginning. In March 2024, researchers published a paper in the New England Journal of Medicine that elevated concerns to an entirely new level. They examined patients who had undergone surgery to remove plaque from their carotid arteries, the major blood vessels that supply the brain. What they found was startling. Patients who had microplastics embedded in their arterial plaque had a significantly higher risk of heart attack, stroke, and death compared to those without plastic contamination. This was one of the first studies to directly link microplastics to specific health outcomes in humans, and the implications were profound. Then came the brain studies. In early 2024, researchers at the University of New Mexico made perhaps the most disturbing discovery yet. They examined brain tissue from deceased individuals and found that not only were microplastics present, but the concentrations in brain tissue were substantially higher than in the liver or kidneys. The median concentration in brain samples from 2024 was nearly 5,000 micrograms per gram of tissue. To put that in perspective, that's roughly half of 1% of your brain tissue. By weight, that is now plastic. And here's what might be the most concerning finding. When researchers compared brain samples from people who died in 2016 versus those who died in 2024, they found that the concentration of microplastics in the brain had increased significantly in just 8 years. The plastic pollution in our bodies isn't static. It's accelerating. We're accumulating more plastic particles faster with each passing year. These discoveries force scientists to confront an uncomfortable truth. The human body has become a repository for synthetic materials that didn't exist in nature until we created them. Every organ system in your body now contains plastic. Your thyroid gland, your testicles or ovaries, your heart muscle, your placenta if you're pregnant. Even the meconium, the first stool of newborn babies, contains microplastics. We are, as one researcher put it, born pre-polluted. The implications of this are staggering. Microplastics have been linked to inflammation, oxidative stress, and cellular damage in laboratory studies. They've been associated with changes in metabolic function, disruptions to the immune system, and alterations in gene expression. Animal studies have shown that exposure to microplastics can impair reproduction, affect brain development, and increase susceptibility to disease. But the full extent of what these particles are doing inside human bodies remains largely unknown. We're in the early stages of understanding a phenomenon that may be reshaping human health on a global scale. This is a story about how the most successful material ever invented, plastic, became one of the most pervasive pollutants in human history. It's about how tiny fragments of this material, invisible to the naked eye, have infiltrated biological systems that evolved over millions of years to protect us from external threats. It's about how something we can't see, can't taste, and barely knew existed until recently may be silently affecting our health in ways we're only beginning to comprehend. The story starts with understanding what these particles actually are, where they come from, and how they manage to invade the most protected tissues in our bodies. To understand the threat, we need to understand the invader. When most people think of plastic pollution, they picture bottles washing up on beaches, grocery bags caught in trees, or the notorious Great Pacific Garbage Patch, that vast accumulation of plastic debris floating in the ocean. But the real story of plastic pollution isn't about what we can see, it's about what we can't. Microplastics are generally defined as plastic particles smaller than 5 millimeters in size. To give you a sense of scale, 5 millimeters is about the size of a sesame seed, but many microplastics are far smaller than that. Some are measured in micrometers, millionths of a meter, making them smaller than the width of a human hair. And then there are nanoplastics, particles smaller than 1 micrometer, so tiny they can only be seen with powerful electron microscopes. These are the particles that concern scientists most, because their minute size allows them to penetrate cells and cross biological barriers that larger particles cannot breach. The story of how plastic fragments became ubiquitous in our environment begins with understanding that plastic is essentially immortal. When you throw away a plastic bottle, it doesn't decompose the way organic materials do. There are no bacteria or fungi that have evolved to efficiently break down the synthetic polymers we've created. Instead, plastic breaks down through mechanical processes, weathering under sun, wind, and water, fragmenting into smaller and smaller pieces. But it never truly disappears. Every molecule of plastic ever manufactured, except for the small fraction that has been incinerated, still exists somewhere on this planet in some form. Scientists divide microplastics into 2 categories based on their origin: primary and secondary. Primary microplastics are intentionally manufactured at small sizes. These include the microbeads that were added to cosmetics and personal care products, those tiny plastic spheres in exfoliating face washes and toothpastes. Although many countries have now banned plastic microbeads in personal care products, they were used extensively for years, and billions of these particles were washed down drains and into waterways. A single use of toothpaste during brushing could release approximately 4,000 microbeads. Multiply that by millions of people using these products daily for years, and you begin to understand the scale. Primary microplastics also include plastic pellets called nurdles, the small, round, lens-shaped pieces of raw plastic that are the building blocks of the plastics industry. These pellets are melted down and moulded into all the plastic products we use, but nurdles are frequently spilled during transportation and manufacturing, and once they enter the environment, they persist indefinitely. Beaches near plastics, manufacturing facilities can be carpeted with these tiny pellets, which are then eaten by seabirds and marine life that mistake them for food. But primary microplastics are dwarfed in quantity by secondary microplastics, particles that form from the breakdown of larger plastic items. This is where the real invasion begins. Every plastic item you've ever used is slowly fragmenting into microplastics. The plastic water bottle you drank from this morning is shedding particles. The synthetic fibres in your clothing are releasing microplastics with every wash. The plastic cutting board in your kitchen is abrading into tiny fragments every time you cut vegetables. Car tyres, which contain synthetic rubber polymers, shed microplastic particles onto roads with every rotation. These tyre particles are then blown into the air or washed into storm drains, eventually finding their way into soil, water, and the air we breathe. The composition of microplastics is as diverse as the plastics industry itself. The most common types found in environmental samples include polyethylene, used in shopping bags and bottles, polypropylene, used in packaging and containers, polyethylene terephthalate, or PET, used in beverage bottles and synthetic fibres, polystyrene, used in food containers and packaging, and polyvinyl chloride, or PVC, used in pipes, flooring, and countless other applications. Each of these polymers has slightly different properties, and each may have different effects when it enters biological systems. But the plastic polymer itself is only part of the story. To understand the true nature of microplastics, you need to understand that plastic is never pure. It's a complex mixture of polymers and additives. These additives are chemicals intentionally added during manufacturing to give plastics their desired properties. Plasticizers make plastic flexible. Flame retardants make it fire-resistant. Antioxidants prevent degradation. Colorants give it appearance. UV stabilizers protect it from sunlight. The crucial fact is that these additives are typically not chemically bonded to the plastic polymer. They're simply mixed in. This means they can leach out over time, especially when exposed to heat, fat, or acids. When you microwave food in a plastic container, additives leach into your food. When you drink hot coffee from a plastic-lined cup, additives leach into your beverage. When a microplastic particle enters your stomach, the acidic environment can cause additives to leach into your digestive system. Among the most concerning additives are bisphenol A, commonly known as BPA, and phthalates. BPA is used to make polycarbonate plastics and epoxy resins, the linings of food and beverage cans. It's an endocrine-disrupting chemical, meaning it can interfere with your body's hormonal systems. Over 92% of Americans have detectable levels of BPA in their urine, reflecting how ubiquitous exposure has become. Phthalates are plasticizers used to make plastic flexible and durable. They're found in hundreds of products, from vinyl flooring to medical tubing to cosmetics. Like BPA, phthalates are endocrine disruptors. They can interfere with testosterone and estrogen function, affecting reproductive development and function. The European Union has classified certain phthalates as substances of very high concern and restricted their use, but they remain widespread in the environment and in products, continually leaching into our bodies. The numbers are staggering. Humans are estimated to inhale or ingest between 74,000 and 121,000 microplastic particles per year through eating, drinking, and breathing. If you drink your required daily water intake exclusively from plastic bottles, you could be ingesting an additional 90,000 microplastic particles per year. A study analysing fecal samples estimated a daily intake of approximately 200 to 330 microplastic particles, translating to roughly 40,000 to 50,000 particles ingested annually through food and beverages alone. But these estimates, alarming as they are, likely underrepresent true exposure. Many studies can only detect microplastics above a certain size threshold, typically around 1 micrometer. Nanoplastics, those particles smaller than 1 micrometer, are much more difficult to detect and quantify. Yet these smallest particles may be the most numerous and potentially the most harmful because they're small enough to cross cell membranes and infiltrate cellular structures. Recent research using advanced detection methods has found that the number of plastic particles in bottled water, for instance, may be 10 to 100 times higher than previous estimates when nanoplastics are included in the count. One study found an average of 240,000 detectable plastic fragments per litre of bottled water, with 90% of these being nanoplastics. These particles are so small that 100 of them lined up would span less than the width of a human hair. The physical characteristics of microplastics vary enormously. They come in different shapes: fibres from synthetic textiles, fragments from degraded plastic items, films from plastic bags, foams from packaging, and spheres from microbeads. Each shape may behave differently in the environment and in biological systems. Fibres, which are among the most common forms found in environmental samples, can be particularly concerning because their elongated shape may make them more likely to penetrate tissues or cause physical damage. The surfaces of microplastics are also not smooth. As plastic weathers and fragments, it develops cracks, pits, and a rough texture. This rough surface increases the particle's surface area, which in turn increases its ability to absorb other pollutants from the environment. Microplastics act like tiny sponges, soaking up heavy metals like lead and mercury, pesticides, polycyclic aromatic hydrocarbons from combustion, and polychlorinated biphenyls, those notorious industrial chemicals that were banned decades ago but persist in the environment. When a microplastic particle enters your body, it's not coming alone. It's bringing with it a cargo of environmental contaminants. What makes this particularly insidious is that microplastics can concentrate these pollutants to levels far higher than what's found in the surrounding environment. The plastic acts as a vector, transporting harmful chemicals directly into organisms and, ultimately, into human tissues. It's like a Trojan horse, smuggling toxins past our body's defences. The production of plastics has increased exponentially since mass production began in the 1950s. In 1950, the world produced about 2 million metric tons of plastic per year. By 2021, that number had risen to nearly 400 million metric tons annually. And with this increase in production has come an increase in plastic pollution. An estimated 10 to 40 million metric tons of microplastics are released into the environment every year. If current trends continue, this number could double by 2040. Every stage of plastic's life cycle generates microplastics. Manufacturing plants release plastic pellets and particles. Consumer use generates microplastics through abrasion and degradation. Washing synthetic textiles releases hundreds of thousands of microplastic fibres per load into wastewater. These fibres are too small to be effectively captured by wastewater treatment plants, so they flow into rivers, lakes, and oceans. Car tyres shed microplastic particles onto roads, where they're washed into storm drains or blown into the air as dust. And here's a crucial point. Once microplastics enter the environment, they accumulate. They don't break down into harmless substances. They simply break into smaller and smaller pieces. This means that every piece of plastic ever made is still out there somewhere, slowly fragmenting, joining the growing burden of plastic pollution that saturates our planet. This brings us to a fundamental question. If microplastics are everywhere in the environment, how exactly do they get inside our bodies? The answer involves 3 primary pathways: what we eat, what we drink, and what we breathe. Your body has evolved elaborate defences against foreign invaders. Your skin is a formidable barrier, largely impermeable to particles and microorganisms. Your respiratory system has mucus and tiny hair-like structures called cilia that trap particles and sweep them out of your airways. Your digestive system has stomach acid that can break down many substances, and a gut lining that carefully controls what enters your bloodstream. But these defences, honed over millions of years of evolution, never encountered synthetic polymers. They were never designed to protect against microplastics. And so, these particles slip through, exploiting vulnerabilities in systems that never evolved to recognise them as threats. The first pathway is ingestion, taking microplastics into your body through food and drink. This is likely the primary route of human exposure. Microplastics have been found in an astounding array of foods. They're in seafood, which makes intuitive sense given the extensive plastic pollution in oceans and the tendency of marine organisms to ingest microplastics. Studies examining fish caught for human consumption have found microplastics in the digestive tracts and tissues of numerous species. When you eat fish or shellfish, you're consuming whatever they've consumed, including microplastics, but seafood is far from the only contaminated food. Microplastics have been detected in honey, where they likely come from atmospheric deposition on flowers or from plastic beekeeping equipment. They've been found in sugar, salt, and beer. A study examining table salt from different countries found microplastic contamination across all samples, with some containing hundreds of particles per kilogram. Every time you salt your food, you may be seasoning it with plastic. Drinking water is a major source of microplastic exposure. Studies have found microplastics in tap water across the globe, from North America to Europe to Asia. The contamination rates are remarkably high. One large-scale study found microplastics in 83% of tap water samples from 14 countries, but bottled water may be even worse. Multiple studies have shown that bottled water typically contains significantly more microplastics than tap water. The plastic bottles themselves shed particles into the water they contain, especially when exposed to heat or stored for extended periods. One study found that a single litre of bottled water could contain hundreds of thousands of plastic particles, with the majority being nanoplastics too small for previous detection methods to identify. The way we prepare and store food also contributes to exposure. Heating food in plastic containers or plastic wrap causes the plastic to shed particles and additives into the food. Microwave-safe plastic is not truly safe from releasing microplastics. It simply means the plastic won't melt at microwave temperatures, but it's still shedding particles, especially with repeated heating. Studies have shown that heating plastic baby bottles to prepare infant formula can release millions of microplastic particles into the liquid. Infants consuming formula from plastic bottles may ingest over 2 million microplastic particles per day. Plastic food packaging is another source. The plastic films used to wrap cheese, meat and other foods can shed particles onto the food surface. Plastic cutting boards release microplastic particles when you cut food on them. Even the plastic bristles of tea bags can release microplastics when steeped in hot water. One study found that a single plastic tea bag steeped in hot water could release over 2 million microplastic particles into your cup. That's millions of plastic particles in a single cup of tea. The second major pathway is inhalation, breathing microplastics into your lungs. This route of exposure has received less attention than the ingestion, but may be equally or more significant. Air, particularly indoor air, contains substantial quantities of microplastic particles. These airborne microplastics come from numerous sources. Synthetic textiles shed microfibers continuously. Every time you sit on a couch covered in synthetic fabric, every time you walk across a carpet made from synthetic fibres, every time you handle a fleece jacket or polyester blanket, tiny fibres are released into the air. These fibres float through your home, settling onto surfaces as dust and remaining suspended in the air you breathe. Studies measuring microplastic concentrations in indoor air have found levels that can reach dozens of particles per cubic metre. When you breathe, you're inhaling these particles. Larger particles are typically trapped in your nose and upper airways and expelled through mucus and coughing. But smaller particles, particularly those below 10 micrometers in size, can penetrate deeper into your lungs, reaching the alveoli, the tiny air sacs where oxygen exchange occurs. And the smallest particles, the nanoplastics, may be able to cross from the alveoli into your bloodstream, providing a direct pathway for plastic particles to circulate throughout your body. One study estimated that humans inhale approximately 39,000 to 52,000 microplastic particles per year through breathing alone. Indoor air exposure likely accounts for a significant portion of this. We spend the majority of our time indoors, and indoor air concentrations of microplastics are often higher than outdoor air because of the concentration of synthetic materials in homes and the tendency for particles to accumulate in enclosed spaces. Outdoor air also contains microplastics, blown up from roads where tyre particles accumulate, lifted from contaminated soils, or carried in atmospheric currents from distant sources. Studies have detected microplastics in atmospheric fallout, the particles that settle from the air onto surfaces. In urban areas, this fallout can reach 29 to 280 particles per square metre per day. Every surface outdoors is being continuously dusted with microscopic plastic particles, and every breath you take outdoors is pulling some of that contaminated air into your lungs. The third pathway is dermal contact, microplastics contacting and potentially penetrating your skin. This route is less well understood and likely less significant than ingestion or inhalation, but it still merits attention. Skin is generally an effective barrier against particles, but nanoplastics, those particles smaller than 100 nanometers, may be small enough to penetrate intact skin. Damaged or broken skin would be even more vulnerable to particle penetration. Personal care products are a potential source of dermal exposure. Although microbeads have been banned in many countries, other plastic-containing ingredients remain in cosmetics, lotions and creams. When you apply these products to your skin, you're potentially exposing yourself to plastic particles. Synthetic fabrics in clothing also shed microfibers, which come into direct contact with skin. Some studies suggest that these fibers might penetrate skin, particularly if the skin is compromised by cuts, abrasions, or conditions like eczema. But regardless of whether microplastics can penetrate healthy skin, dermal contact still leads to ingestion. Your hands touch your face, your mouth, your food. Microplastic particles on your hands are transferred to these surfaces and eventually ingested. Children are particularly vulnerable to this pathway because of their tendency to put their hands and objects in their mouths. Once microplastics enter your body through any of these pathways, they embark on a journey through your biological systems. And this is where the story becomes even more concerning, because research is revealing that microplastics don't simply pass through harmlessly. They accumulate, they persist, and they may be causing damage at every stage of their journey through your body. To understand what microplastics do once they're inside you, we need to follow their path, starting with the most direct route, the digestive system. When you swallow food or drink containing microplastics, you're initiating a complex journey through one of the body's most sophisticated biological systems. The human digestive tract is approximately 30 feet long, beginning at your mouth and ending at your anus, and along this entire route, microplastics can interact with tissues, accumulate in certain areas, and potentially cause harm. The journey begins in your mouth. Studies examining human saliva have not yet widely reported microplastic content, but given the ubiquitous presence of microplastics in food, water and air, it's reasonable to assume that your mouth is regularly exposed to these particles. The oral cavity is where food begins to be broken down mechanically, through chewing and chemically through enzymes in saliva. But microplastics, being synthetic polymers resistant to biological degradation, pass through this stage largely unchanged. They're swallowed along with your food and enter the esophagus. The esophagus is a muscular tube that moves food from your mouth to your stomach through rhythmic contractions called peristalsis. Microplastics follow this same route, propelled by the wave-like muscle movement into your stomach. Here, things get more interesting. The stomach is an acidic environment, with pH levels often dropping below 2, acidic enough to break down proteins and kill most bacteria. But this acid has little effect on plastic polymers. They don't dissolve or degrade in stomach acid. However, the acidic environment can affect the additives in the plastic. Remember those chemicals we discussed, the plasticizers and flame retardants and other additives, mixed into plastic but not chemically bonded to it? Stomach acid can cause these chemicals to leach out of the microplastic particles and into the surrounding gastric fluid. This means that even before the microplastic particles move further into your digestive tract, they may already be releasing toxic chemicals into your stomach. Animal studies have shown that microplastics can accumulate in the stomach, particularly if they're large enough to resist passing through the pyloric sphincter, the valve connecting the stomach to the small intestine. In these cases, the particles may remain in the stomach for extended periods, continuously leaching additives and potentially causing mechanical irritation to the stomach lining. But most microplastics, particularly the smaller ones, pass through the stomach and enter the small intestine. This is where the most significant interactions occur. The small intestine is where the majority of nutrient absorption happens. Its inner surface is covered with millions of tiny finger-like projections called villi, which increase the surface area available for absorption. Each villus is further covered with even tinier projections called microvilli, creating a vast surface area. If you could spread out the entire inner surface of your small intestine, it would cover roughly 30 square meters, about the size of a small studio apartment. This enormous surface area is designed to efficiently absorb nutrients from digested food into your bloodstream. But this same surface area is now exposed to microplastics. Studies in animal models have shown that microplastics can adhere to the intestinal wall, accumulate in the mucus layer that coats the intestine, and potentially penetrate into the intestinal tissue itself. The size of the microplastic particles is crucial here. Larger particles, those above 150 micrometers, are too large to cross the intestinal barrier and are typically excreted in feces. But smaller particles, particularly those below 10 micrometers, may be able to penetrate the intestinal epithelium, the layer of cells that lines the intestine and controls what enters the bloodstream. And nanoplastics, those particles smaller than 1 micrometer, are small enough to be taken up by cells through a process called endocytosis, where the cell membrane engulfs the particle and draws it inside. Once inside intestinal cells, microplastics can cause various types of damage. They can generate reactive oxygen species, highly reactive molecules that cause oxidative stress, a state where damaging reactions occur that overwhelm the cell's antioxidant defenses. This oxidative stress can damage cellular components, including DNA, proteins, and the lipid membranes that form the boundaries of cells and organelles. The result can be inflammation, cellular dysfunction, and even cell death. Studies in cell cultures and animal models have shown that exposure to microplastics triggers inflammatory responses in intestinal cells. The cells release signaling molecules called cytokines that recruit immune cells and amplify the inflammatory response. Chronic inflammation in the intestine is associated with various digestive disorders, including inflammatory bowel disease. There's also evidence that microplastics can disrupt the gut barrier function. The cells lining your intestine are normally connected by tight junctions, protein structures that seal the space between cells and prevent unwanted substances from passing through. Microplastics have been shown to compromise these tight junctions, increasing intestinal permeability. This condition, sometimes called leaky gut, allows bacteria, toxins, and other substances that should stay in the intestinal lumen to cross into the bloodstream, potentially triggering immune responses and contributing to systemic inflammation. The gut microbiome, that vast community of bacteria, viruses, and other microorganisms living in your intestine, is also affected by microplastics. Your microbiome plays crucial roles in digestion, immune function, and even mental health. Studies have shown that exposure to microplastics can alter the composition of the gut microbiome, changing the relative abundance of different bacterial species. Some studies have found that microplastic exposure leads to an increase in potentially harmful bacteria and a decrease in beneficial species. The long-term health consequences of these microbiome alterations are still being investigated, but given the microbiome's importance to overall health, any disruption is concerning. Not all microplastics that enter the intestine are absorbed into tissues or cells. Many pass through and are excreted in feces. Studies analysing human stool samples have consistently found microplastics, with typical findings ranging from a few particles to dozens of particles per gram of feces. This provides evidence that humans are continuously exposed to and excreting microplastics. But the fact that we're excreting them doesn't mean they're harmless. Even particles that are eventually excreted may cause damage during their transit through the digestive system. The large intestine, or colon, is the final stage of the digestive journey. Here, water is absorbed from digested food, and the remaining material is formed into feces. Microplastics that made it through the small intestine end up in the colon, where they may accumulate before being excreted. There's emerging evidence linking microplastic exposure to colorectal health issues. Studies have found higher concentrations of microplastics in tissue samples from patients with inflammatory bowel disease and colorectal cancer compared to healthy controls. This doesn't prove that microplastics cause these conditions, but it suggests a possible association that warrants further investigation. It's important to note that not all microplastics that enter the digestive system stay there. Some of the smallest particles, particularly nanoplastics, can cross the intestinal barrier and enter the bloodstream. And once in the bloodstream, these particles gain access to the entire body. They can be transported to any organ, any tissue, anywhere blood flows, and blood flows everywhere. This brings us to one of the most concerning discoveries in microplastics research. The finding that microplastics circulate in human blood and accumulate in vital organs throughout the body. Your circulatory system is your body's transportation network, delivering oxygen and nutrients to every cell while removing waste products. Your heart pumps approximately 5 litres of blood through an intricate network of arteries, veins and capillaries, vessels so tiny that blood cells must squeeze through single file. This system touches every part of your body, which means that anything circulating in your blood has access to every organ, every tissue, potentially every cell. The discovery in 2022 that microplastics were present in human blood was a watershed moment. Published in the journal Environment International, the study examined blood samples from 22 healthy adult volunteers in the Netherlands. The researchers found plastic particles in 17 of the 22 samples, a detection rate of 77%. The concentrations range from 0.3 to 1.9 micrograms per milliliter of blood. The most common polymer detected was polyethylene terephthalate, better known as PET, followed by polyethylene and polystyrene. These aren't enormous concentrations, but their very presence is significant. Your blood is supposed to be sterile, free of microorganisms and foreign particles. Yet here were synthetic polymers, circulating in the bloodstream of most people tested. And this was unlikely to be an isolated phenomenon limited to the Netherlands. Follow-up studies in other countries have reported similar findings. How do microplastics get into the bloodstream? The primary route appears to be crossing from the gut or lungs into the blood. In the intestine, as we discussed, the smallest particles can be taken up by intestinal cells and transported across the epithelial barrier into the blood vessels that line the intestinal wall. In the lungs, particles that reach the alveoli, the tiny air sacs where gas exchange occurs, can potentially cross into the pulmonary capillaries, the tiny blood vessels surrounding the alveoli. The barrier between the air in your lungs and the blood in these capillaries is extremely thin, designed to allow oxygen and carbon dioxide to pass freely, but this thinness also means it's more permeable to small particles. Nanoplastics with their minute size may be able to slip through. Once in the bloodstream, microplastics are transported throughout the body by blood flow. But where do they go? What organs do they accumulate in? This is where recent research has yielded deeply troubling findings. Studies have detected microplastics in multiple organs beyond the digestive system. They've been found in the lungs, which makes sense given that inhalation is a primary exposure route. But they've also been found in the liver, the body's primary detoxification organ, in the kidneys, which filter blood to remove waste products, in the spleen, part of the immune system. They've been detected in heart tissue, in the heart muscle itself, and perhaps most alarmingly, they've been found in the brain. Let's focus first on the cardiovascular system, the heart and blood vessels. The heart is a muscle that contracts approximately 100,000 times per day, pumping blood throughout your body. Any substance in the blood passes through the heart repeatedly. Over a lifetime, that's billions of passages. If microplastics are accumulating in heart tissue, what effect might this have? The study, published in the New England Journal of Medicine in March 2024, provided some of the first evidence linking microplastics to cardiovascular outcomes in humans. Researchers examined patients undergoing carotid endarterectomy, a surgery to remove plaque buildup from the carotid arteries, the major blood vessels supplying blood to the brain. The surgical procedure involves removing the inner lining of the artery along with the accumulated plaque. This gave researchers access to actual arterial tissue from living patients. When they analysed the removed plaque using advanced techniques, they detected microplastics in about 60% of the samples. The plastics found were primarily polyethylene and polyvinyl chloride. But finding microplastics in arterial plaque wasn't the most significant finding. What made this study groundbreaking was that the researchers followed these patients after their surgery, tracking health outcomes over more than 2 years. They found that patients who had microplastics detected in their carotid plaque had a substantially higher risk of cardiovascular events compared to patients whose plaque did not contain detectable microplastics. Specifically, patients with microplastics had 4.5 times higher risk of heart attack, stroke, or death from any cause over the follow-up period. This was after accounting for other cardiovascular risk factors like smoking, diabetes, and high blood pressure. This study was observational, meaning it can show association but not necessarily causation. It's possible that some other factor associated with microplastic exposure is the actual cause of the increased risk, but the finding is nonetheless concerning and has spurred increased research into the cardiovascular effects of microplastics. How might microplastics contribute to cardiovascular disease? Several mechanisms are possible. First, the physical presence of particles in arterial walls might promote inflammation, contributing to the atherosclerotic process where arteries become hardened and narrowed by plaque buildup. Studies have shown that microplastics can trigger inflammatory responses in cells, releasing pro-inflammatory cytokines that promote atherosclerosis. Second, microplastics might make plaque less stable. Stable plaque, while it narrows arteries, tends to remain in place. Unstable plaque is more likely to rupture, causing a clot that can block blood flow and trigger a heart attack or stroke. Some researchers hypothesize that microplastics embedded in plaque might make it more prone to rupture. Third, additives leaching from microplastics might have direct cardiovascular effects. Some phthalates, for instance, have been associated with increased blood pressure and changes in cholesterol levels. BPA has been linked to endothelial dysfunction, impairment of the cells lining blood vessels, which is an early step in cardiovascular disease development. But the cardiovascular system isn't the only area of concern. Microplastics are being found throughout the body, and nowhere is this more alarming than in the brain. Of all the organs in your body, your brain is perhaps the most protected. It's encased in your skull, cushioned by cerebrospinal fluid, and shielded by the blood-brain barrier, a selective barrier formed by specialized cells that line the brain's blood vessels. This barrier carefully regulates what substances can pass from your blood into your brain tissue, protecting this most delicate and vital organ from toxins, pathogens, and other threats. But this barrier is not impermeable to microplastics, and recent research has revealed that plastic particles are accumulating in human brain tissue at concentrations that exceed what's found in other organs. The study published in 2024 by researchers at the University of New Mexico represents one of the most comprehensive examinations of microplastics in human organs to date. The researchers obtained samples of kidney, liver, and brain tissue from the New Mexico Office of Medical Investigator, analysing tissues from deceased individuals. Using a technique called pyrolysis gas chromatography mass spectrometry, they were able to detect and quantify plastic particles down to about 200 nanometers in size. The findings were stark. Microplastics were present in all organ types examined, but brain samples exhibited substantially higher concentrations than liver or kidney. The median concentration of microplastics in brain tissue from 2024 samples was approximately 4,900 micrograms per gram of tissue. To put that in context, roughly half of 1% of brain tissue by weight consisted of plastic particles. The predominant plastic type found in brain tissue was polyethylene, the plastic used in shopping bags, food containers, and countless other applications. But the researchers also found polystyrene, polyvinyl chloride, and various other polymers. Using electron microscopy, they examined the physical characteristics of the particles. Most appeared as irregular, shard-like fragments, suggesting they came from the breakdown of larger plastic items rather than being manufactured as microparticles. Perhaps the most concerning finding was the temporal trend. When the researchers compared brain samples from individuals who died in 2016 versus those who died in 2024, they found that microplastic concentrations had increased significantly over this 8-year period. The 2024 samples contained approximately 50% higher concentrations than the 2016 samples. This suggests that microplastic pollution in human brain tissue is not static, but accelerating. How do microplastics cross the blood-brain barrier and enter brain tissue? This is an area of active research and the mechanisms are not fully understood. The blood-brain barrier is formed by endothelial cells that line brain capillaries, cells connected by extremely tight junctions that prevent most large molecules and particles from passing through. But nanoplastics, with their tiny size, may be small enough to slip through gaps in these junctions or be actively transported across by cellular mechanisms. One hypothesis involves a process called transcytosis, where particles are taken up by endothelial cells on the blood side, transported through the cell in vesicles, and released on the brain side. This process normally allows certain necessary substances to cross the barrier, but it might also inadvertently transport nanoplastics. Another possibility is that microplastics somehow disrupt the tight junctions between endothelial cells, creating larger gaps through which particles can pass. Once inside the brain, microplastics encounter an environment unlike any other in the body. The brain contains approximately 86 billion neurons, the specialized cells that process and transmit information through electrical and chemical signals. It also contains supporting cells called glia, which outnumber neurons and play crucial roles in brain function. The brain is the seat of consciousness, memory, emotion, and thought. It controls every function of your body. Any disruption to brain function can have profound consequences. Animal studies have provided evidence that microplastics can accumulate in brain tissue and cause neurological changes. Studies in mice have shown that when pregnant mice are exposed to microplastics, the particles can cross the placental barrier and accumulate in fetal brain tissue. Offspring of these exposed mice show behavioral changes including anxiety-like behavior and altered social interactions. The plastic particles have been found specifically in the thalamus, a brain region involved in sensory processing and sleep regulation. Other animal studies have demonstrated that microplastic exposure can cause oxidative stress in the brain, inflammation, and changes in the levels of neurotransmitters, the chemical messengers that neurons use to communicate. Some studies have found that microplastics can disrupt the function of microglia, the brain's immune cells. Microglia normally protect the brain by removing damaged cells and fighting infections, but exposure to microplastics can cause them to become overactivated, releasing inflammatory molecules that can damage neurons. A particularly concerning study published in early 2025 found that microplastics in the bloodstream can induce cerebral thrombosis, the formation of blood clots in brain blood vessels, by causing cell obstruction. In experimental animals, this led to neurobehavioral abnormalities. If similar effects occur in humans, it could have implications for stroke risk and neurological function. There's also growing concern about whether microplastics might contribute to neurodegenerative diseases. Alzheimer's disease, Parkinson's disease, and other neurodegenerative conditions involve the accumulation of abnormal proteins in the brain and progressive loss of neurons. While these diseases are multifactorial, involving genetic, environmental, and lifestyle factors, chronic inflammation and oxidative stress are believed to play important roles in their development and progression. If microplastics induce inflammation and oxidative stress in the brain, as animal studies suggest, they could potentially contribute to or accelerate neurodegenerative processes. Some researchers have begun to investigate whether there's a correlation between microplastic exposure and the risk of neurodegenerative diseases, but this research is in its early stages. It's a difficult question to answer because neurodegenerative diseases develop over decades. and we've only recently become aware of widespread human exposure to microplastics. Definitive answers may not come for years. Beyond specific diseases, there's the question of whether microplastics affect cognitive function more broadly. Could they impair memory, attention, or learning? Could they affect mood or behaviour? These are questions that urgently need investigation, but are challenging to study because of the difficulty in measuring microplastic exposure in living individuals, and because any effects are likely to be subtle and develop over long periods. Children may be particularly vulnerable to the effects of microplastics on brain development. The developing brain undergoes critical periods of rapid growth and organisation, times when it's particularly sensitive to environmental influences. Exposure during these critical periods can have lasting effects. Studies have shown that children are exposed to microplastics even before birth, as the particles can cross the placental barrier and through breast milk after birth. What the long-term consequences of this early exposure might be is unknown, but it's a question that concerns many researchers and parents. The brain findings are made more alarming by the fact that microplastics don't arrive in the brain alone. They bring with them the chemical additives embedded in the plastic and the environmental pollutants adsorbed onto their surfaces, and these chemicals may be even more dangerous than the plastic particles themselves. When you think about microplastics as a threat to human health, it's tempting to focus on the physical particles themselves, the tiny fragments of plastic lodged in your tissues. But in many ways, the plastic polymer is just the delivery vehicle. The real threat may be the chemicals that come along for the ride. As we've discussed, plastic is never pure. It's a complex mixture of polymers and additives. These additives include plasticizers that make plastic flexible, flame retardants that make it fire-resistant, stabilizers that protect it from degradation, and numerous other substances that give plastic its desired properties. Globally, more than 13,000 different chemicals are associated with plastic production and use. Of these, over 3,200 have been identified as substances of potential concern based on their known hazardous properties. The crucial fact is that most additives are not chemically bonded to the plastic polymer. They're simply mixed in during manufacturing. This means they can leach out over time, especially when exposed to heat, solvents, fats, or acids. Every plastic item is slowly releasing chemicals into whatever it contacts. Bisphenol A, commonly known as BPA, is perhaps the most well-known plastic additive. It's used to make polycarbonate plastics and epoxy resins. You've likely heard of BPA because public awareness campaigns and regulatory actions have highlighted its potential health risks, leading to BPA-free labelling on many products. But BPA is far from gone. It's still used in numerous applications, and even BPA-free products often contain chemical relatives like bisphenol S or bisphenol F, which have similar structures and may have similar effects. BPA is what's called an endocrine disrupting chemical. It can mimic the hormone estrogen, binding to estrogen receptors in cells and triggering responses that estrogen would normally trigger. This can disrupt normal hormonal signaling, affecting development, reproduction, and metabolism. Even at low doses, exposure during critical developmental periods can have lasting effects. Studies have linked BPA exposure to a wide range of health problems. In animal studies, BPA has been shown to affect brain development, alter behaviour, disrupt thyroid function, and interfere with reproductive development. In humans, BPA exposure has been associated with increased risk of obesity, cardiovascular disease, diabetes, and reproductive disorders. A 2023 reassessment by the European Food Safety Authority dramatically lowered the recommended tolerable daily intake for BPA by 20,000-fold, based on new evidence of health risks at much lower doses than previously thought. The ubiquity of BPA exposure is striking. Studies by the US Centers for Disease Control and Prevention have found detectable levels of BPA in the urine of over 92% of Americans tested. This reflects constant ongoing exposure through multiple sources, and because BPA has a relatively short half-life in the body, around 6 hours, The fact that it's constantly detectable means people are being continuously exposed. Phthalates are another major category of plastic additives. These chemicals are used as plasticizers to make plastic soft and flexible. They're found in vinyl flooring, medical tubing, food packaging, personal care products, and numerous other items. There are many different phthalates in use, and some are more toxic than others. But as a class, phthalates are concerning because they're endocrine disruptors that can interfere with hormonal systems, particularly affecting reproduction. Phthalates have been linked to reduced testosterone levels in males, altered timing of puberty, reduced sperm quality, and various reproductive abnormalities. In women, phthalate exposure has been associated with altered hormone levels and increased risk of pregnancy complications. There's also evidence linking phthalates to metabolic disorders, including obesity and insulin resistance. Like BPA, phthalates are ubiquitous. They've been detected in the urine of nearly all people tested in biomonitoring studies. They're in house dust, in indoor air, in food, and in drinking water. And like BPA, their presence in microplastics means they're being transported directly into human tissues. The concerning thing about microplastics as vehicles for these additives is that they can deliver concentrated doses. A microplastic particle embedded in tissue is like a slow-release capsule, continuously leaching additives into the surrounding tissue. The pH, temperature, and chemical environment of different tissues can affect the rate of leaching. In the acidic environment of the stomach, or in fatty tissues that can dissolve lipophilic chemicals, leaching may be particularly efficient. But additives are only part of the chemical story. Microplastics also act as vectors for environmental pollutants. Their surfaces can absorb various toxic compounds from the environment, often concentrating these pollutants to levels far exceeding what's found in the surrounding water or soil. Persistent organic pollutants, or POPs, are chemicals that resist degradation and accumulate in the environment and in living organisms. They include polychlorinated biphenyls, or PCBs, industrial chemicals that were banned decades ago but persist in the environment, polybrominated diphenyl ethers, or PBDEs, flame retardants that have also been phased out but remain widespread, and various pesticides and other compounds. Many POPs are known carcinogens or endocrine disruptors, and they readily adsorb onto the surfaces of microplastics. Polycyclic aromatic hydrocarbons, or PAHs, are another class of environmental pollutants that microplastics can carry. PAHs are formed during incomplete combustion of organic matter and are present in air pollution, wildfire smoke, and contaminated soils. Many PAHs are carcinogenic. Studies have shown that PAHs readily adsorb onto microplastics and can be released once the particles enter biological systems. Heavy metals like lead, cadmium, and mercury can also adsorb onto microplastics. These metals are toxic to numerous organ systems and can accumulate in the body over time. Microplastics may facilitate the transport of these metals into organisms and into human tissues. What makes this particularly insidious is the concept of the cocktail effect. When you're exposed to multiple chemicals simultaneously, they can interact in complex ways. Some chemicals may enhance the toxicity of others through synergistic effects. A dose of chemical A alone and a dose of chemical B alone might each be below the threshold for causing harm, but together they might have significant effects, and microplastics often carry multiple chemicals, creating complex mixtures of pollutants delivered together. Research into these combined effects is challenging because of the sheer number of possible combinations and the difficulty of testing them all. But studies that have examined co-exposure to microplastics and environmental pollutants consistently show that the combined toxicity is often greater than would be predicted from the sum of the individual toxicities. There's also the issue of biofilms. Microplastics in the environment often become coated with biofilms, layers of microorganisms including bacteria, algae, and fungi that colonize the particle surfaces. These biofilms can include pathogenic bacteria, potentially making microplastics vectors for disease. When a microplastic particle carrying a biofilm enters your body, It's bringing with it a community of microorganisms that may include harmful species. One particularly concerning aspect is the potential for antibiotic-resistant bacteria to colonize microplastics. Antibiotic resistance is already a major public health threat, and there's evidence that microplastics in the environment can harbour antibiotic-resistant bacteria and genes encoding antibiotic resistance. If these particles enter your body, they could potentially transfer resistant bacteria or resistance genes to your own microbiome, contributing to the spread of antibiotic resistance. The cumulative effect of all these chemical exposures through microplastics is difficult to quantify, but potentially profound. Your body has detoxification systems, primarily in the liver, that work to break down and eliminate toxic compounds. But these systems evolved to deal with natural toxins, substances that existed in the environment our ancestors encountered. They weren't designed to handle the flood of synthetic chemicals we're now exposed to, and the exposure is continuous. Unlike a one-time exposure to a toxin that your body can process and eliminate, microplastic exposure is constant and lifelong. Every day you're ingesting, inhaling, and possibly absorbing through your skin more microplastic particles carrying more additives and pollutants. The particles may be excreted or may accumulate, but either way you're being continuously re-exposed. It's like trying to bail out a boat while someone is continuously pouring water into it. This brings us to one of the most concerning aspects of microplastic exposure: the effects on reproduction and development. If there's one system where the effects of microplastics are particularly alarming, it's the reproductive system. Evidence is mounting that microplastics and their associated chemicals can disrupt reproduction at multiple levels, from affecting fertility to altering development of offspring to potentially passing effects across generations. Let's start with male reproduction. In May 2024, researchers published a study that sent shockwaves through the scientific community and the public. They examined testicular tissue from deceased individuals and found microplastics in every single sample. The concentrations were substantial, ranging from about 120 to 330 micrograms per gram of tissue. For comparison, these were higher than the concentrations found in blood, but lower than those found in brain tissue in other studies. The most common plastic found in testicular tissue was polyethylene, followed by PVC. And here's where it gets particularly concerning. The researchers also examined testicular tissue from dogs, and while microplastics were found in all dog samples as well, the concentrations in human tissue were substantially higher. The dog samples also showed a correlation between the level of PVC and decreased sperm count. How do microplastics affect male fertility? Multiple mechanisms appear to be at play. First, the physical presence of plastic particles in testicular tissue may cause inflammation and oxidative stress, disrupting the delicate environment needed for sperm production. The testes have a unique structure with the blood-testis barrier that protects developing sperm cells from harmful substances in the blood. But this barrier is not impermeable to small particles, and nanoplastics may be able to cross it. Second, the chemicals associated with microplastics can directly interfere with hormone production and function. Phthalates, for instance, are known to be anti-androgenic, meaning they can block or reduce the effects of male hormones like testosterone. Studies have shown that phthalate exposure can reduce testosterone production, disrupt the development of male reproductive organs during fetal development, and impair sperm quality in adults. Third, microplastics can interfere with spermatogenesis, the process by which sperm cells are produced. This process involves the intricate coordination of multiple cell types and is highly sensitive to disruption. Animal studies have shown that exposure to microplastics can reduce sperm count, decrease sperm motility, increase the percentage of abnormal sperm, and cause DNA damage in sperm cells. Sperm DNA damage is particularly concerning because it can lead to infertility, miscarriage, or developmental problems in offspring. Human studies are beginning to corroborate these animal findings. In 2023, researchers reported detecting microplastics in human semen samples. They found particles in 6 out of 10 samples examined, with concentrations ranging from 2 to 12 microplastic particles per milliliter. While the sample size was small, this was the first direct evidence of microplastics in human semen, confirming that these particles can reach the male reproductive tract. There's also troubling evidence of declining sperm counts globally. Multiple studies over the past decades have documented that sperm counts in men in Western countries have declined by more than 50% since the 1970s. The causes of this decline are likely multifactorial, involving lifestyle factors, environmental exposures, and chemical pollutants. But microplastics and their associated chemicals have emerged as prime suspects. Female reproduction is equally affected. Microplastics have been found in ovarian tissue, in the endometrium, the lining of the uterus, and in the placenta. Studies in female animals have shown that microplastic exposure can disrupt the hypothalamic pituitary ovarian axis, the hormonal control system that regulates ovulation and menstruation. This can lead to irregular menstrual cycles, anovulation, and reduced fertility. Animal studies have demonstrated that microplastic exposure can reduce the number of viable ovarian follicles, the structures that contain developing egg cells. A female is born with a finite number of follicles, and once they're depleted, she can no longer conceive. If microplastics accelerate the loss of follicles or interfere with their development, this could affect fertility and potentially bring on earlier menopause. The chemicals in microplastics can also disrupt the hormonal balance needed for reproduction. Estrogen and progesterone must be precisely regulated throughout the menstrual cycle for successful ovulation, conception, and pregnancy. Endocrine disrupting chemicals like BPA and phthalates can interfere with this delicate balance. Studies have found associations between phthalate exposure and altered hormone levels, reduced fertility, and increased risk of pregnancy complications. But perhaps the most concerning findings involve pregnancy and fetal development. Multiple studies have now detected microplastics in human placentas. The placenta is the organ that forms during pregnancy to nourish the developing fetus, exchanging nutrients, oxygen, and waste between maternal and fetal blood. It's also supposed to act as a barrier, protecting the fetus from harmful substances. Yet microplastics are crossing this barrier. A groundbreaking study published in 2021 was the first to report microplastics in human placentas. Researchers examined 6 placentas from healthy pregnancies and found microplastic particles in 4 of them, with a total of 12 particles identified. The particles were found on both the maternal side and fetal side of the placenta, as well as in the chorioamniotic membranes that surround the fetus. The plastics were primarily polyethylene and polypropylene. Since then, multiple other studies have confirmed the presence of microplastics in placentas, with some finding particles in every placenta examined. One study from 2024 found microplastics not just in the placenta, but also in fetal cord blood and in meconium, the first stool of newborn babies. This proves that microplastics are not only reaching the placenta, but crossing into the fetal circulation and accumulating in fetal tissues. What are the consequences of fetal exposure to microplastics? Animal studies provide concerning clues. When pregnant mice or rats are exposed to microplastics, their offspring show various abnormalities. These include altered growth patterns, with some studies finding reduced birth weight, and others finding increased susceptibility to obesity later in life. Behavioral changes are common, with offspring showing increased anxiety-like behavior, altered social interactions, and in some cases features resembling autism spectrum disorders. Some of these effects persist across multiple generations. Studies exposing pregnant animals to plastic additives like BPA and phthalates have found that abnormalities in reproductive organ development and function appear not just in the directly exposed offspring but in subsequent generations, even though those later generations weren't directly exposed. This suggests epigenetic effects, changes in how genes are expressed without changes to the DNA sequence itself, that can be passed from parent to offspring. The implications for human health are profound. If microplastic exposure during pregnancy can alter fetal development, affect hormone systems, or change brain structure and function, we may be witnessing the emergence of developmental effects that will only become fully apparent as today's children grow up. Some researchers have begun to question whether microplastic exposure might be contributing to observed increases in developmental disorders, neurodevelopmental problems, early puberty, or reproductive issues in younger generations. Breast milk, that perfect food for infants, has also been found to contain microplastics. Studies have detected plastic particles in human breast milk from mothers in multiple countries. While breast milk remains highly beneficial for infant health overall, the presence of microplastics raises questions about infant exposure during this critical period of rapid growth and development. Infant formula prepared in plastic bottles may be an even greater source of exposure. Studies have shown that shaking and heating formula in plastic bottles can release millions of microplastic particles into the liquid. Infants consuming formula from plastic bottles may be exposed to over 2 million microplastic particles per day. This exposure is particularly concerning because infants' organs and systems are still developing, and their smaller body size means that the same number of particles represents a higher dose per kilogram of body weight compared to adults. The reproductive effects of microplastics aren't limited to the exposed individual. Through effects on fertility, pregnancy, and fetal development, microplastics have the potential to affect future generations. And this raises an uncomfortable question. Are we witnessing the beginning of a long-term impact on human reproduction and development that will only become fully apparent decades from now? While the reproductive system bears a heavy burden from microplastic exposure, another critical system is also under assault: the immune system, your body's defense against disease. Your immune system is your body's defensive army, a complex network of cells, tissues, and organs that work together to identify and neutralize threats from bacteria, viruses, parasites, and cancerous cells. It's been honed by millions of years of evolution to recognize the difference between self and non-self, between what belongs in your body and what should be destroyed. But microplastics present a novel challenge, something your immune system never evolved to handle. When immune cells encounter microplastics, they don't quite know what to do. These particles aren't alive like bacteria or viruses, but they're clearly foreign. They're not supposed to be there. And so the immune system responds, but that response can become problematic when the threat is continuous and can't be eliminated. Let's start with what happens when an immune cell encounters a microplastic particle. Macrophages, large white blood cells that patrol your tissues looking for threats, are particularly important in the immune response to microplastics. When a macrophage encounters a foreign particle, it normally engulfs it through a process called phagocytosis, cellular eating. The particle is enclosed in a membrane-bound compartment inside the cell, where it can be broken down by digestive enzymes. But microplastics are synthetic polymers that resist biological degradation. A macrophage can engulf a microplastic particle, but it can't break it down. The particle remains intact inside the cell. Studies have shown that this can trigger a cascade of cellular stress responses. The macrophage generates reactive oxygen species, those highly reactive molecules we've discussed before, in an attempt to destroy the particle. But this oxidative burst ends up damaging the macrophage's own cellular components. The frustrated macrophage also releases inflammatory signaling molecules called cytokines. These include interleukin-6, interleukin-1 beta, and tumor necrosis factor alpha, molecules that recruit other immune cells and amplify the inflammatory response. In a normal immune response against a bacteria or virus, this inflammation is beneficial, helping to contain and eliminate the threat. But when the trigger is a microplastic particle that can't be eliminated, the inflammation becomes chronic. Chronic inflammation is linked to numerous health problems. It contributes to cardiovascular disease, accelerates aging, promotes cancer development, and is implicated in autoimmune diseases where the immune system mistakenly attacks the body's own tissues. If microplastics are causing widespread, low-level chronic inflammation throughout the body, this could have far-reaching health consequences. Studies in cell cultures and animal models have consistently shown that microplastic exposure triggers inflammatory responses. In studies with intestinal cells, microplastic exposure leads to increased production of pro-inflammatory cytokines and activation of inflammatory signaling pathways. In studies with lung cells, inhaled microplastics cause similar responses. Animal studies have shown that microplastic exposure can lead to inflammation in multiple organs, including the liver, kidneys, intestines, and lungs. But the immune response to microplastics isn't just about inflammation. There's also evidence that microplastics can directly damage immune cells. Neutrophils, white blood cells that are part of the first line of defense against infection, have been shown to undergo cell death after ingesting microplastics. When neutrophils die, they can release their DNA in a process called net formation, neutrophil extracellular traps, where DNA is expelled from the cell to trap pathogens. While this can be a useful defense mechanism, excessive net formation is associated with tissue damage and autoimmune diseases. Microplastics can also affect the adaptive immune system, the component that provides targeted, long-lasting immunity to specific pathogens. This system involves T cells and B cells, lymphocytes that recognize specific antigens. Some studies have found that microplastic exposure alters the ratios of different T cell subtypes, potentially compromising immune function. There's also concerning evidence that microplastics might affect immune development in young animals. The immune system undergoes critical development during infancy and childhood, and exposure to microplastics during these periods might alter how the immune system matures. Animal studies have shown that offspring of parents exposed to microplastics during pregnancy and lactation show altered immune responses and changes in immune cell populations. The gut immune system deserves special attention because the gut contains more immune cells than any other part of the body. Approximately 70% of your immune system is associated with your digestive tract, where immune cells must balance tolerating beneficial bacteria and food antigens while defending against pathogens. Microplastics entering through the digestive system have direct contact with this extensive immune network. Studies have shown that microplastics in the gut can disrupt immune homeostasis, the balance between tolerance and active immune responses. They can cause inflammatory changes in the intestinal lining, alter the composition of gut-associated immune cells, and potentially contribute to conditions like inflammatory bowel disease. In patients with inflammatory bowel disease, some studies have found higher concentrations of microplastics in intestinal tissue compared to healthy controls, suggesting a possible link between microplastic exposure and disease severity. The gut microbiome, that vast community of microorganisms in your intestine, is intimately connected to immune function. Your immune system and your microbiome are in constant communication, and a healthy microbiome is essential for proper immune development and function. As we discussed earlier, microplastics can disrupt the gut microbiome, changing the balance of bacterial species. This disruption can have cascading effects on immune function. There's also the possibility that microplastics might make people more susceptible to infections or less able to mount effective immune responses to vaccines. Some animal studies have shown that microplastic exposure can reduce the effectiveness of immune responses to pathogens or vaccines, though this hasn't been definitively demonstrated in humans yet. A study examining the effects of microplastics on COVID-19 infection in mice found that microplastic exposure altered the immune response to the virus, suppressing innate immunity in the early stages of infection and increasing pro-inflammatory responses later. Another concerning aspect is the potential for microplastics to contribute to autoimmune diseases, conditions where the immune system attacks the body's own tissues. The chronic inflammation caused by microplastics, combined with potential effects on immune regulation, could theoretically increase the risk of autoimmunity, though this hasn't been proven in humans. It's a hypothesis that warrants investigation. Allergic diseases are also a consideration. Some studies have suggested that microplastic exposure might promote allergic responses, potentially contributing to the rising prevalence of allergies and asthma in developed countries. The mechanisms aren't clear, but could involve effects on immune cell development, changes in the microbiome, or direct inflammatory effects of particles in airways. It's important to note that much of the evidence for immune effects comes from cell culture studies and animal experiments using exposure levels that may be higher than typical human exposure. Translating these findings to real-world human health impacts is challenging, but the consistency of findings across multiple studies and species suggests that the effects are real and worth taking seriously. The immune system effects of microplastics are particularly concerning because the immune system touches every aspect of health. Immune dysfunction can manifest as increased susceptibility to infections, slower wound healing, increased cancer risk, autoimmune disease, chronic inflammation contributing to cardiovascular disease and metabolic disorders, and potentially accelerated aging. If microplastics are broadly compromising immune function across populations, the health consequences could be profound and diverse, affecting multiple disease processes. But to fully understand the scope of human exposure to microplastics and where these particles come from, we need to trace their path from the environment to our bodies. The microplastics inside your body didn't originate there. They began their journey in the environment, and that journey often spans continents and oceans, involving complex pathways through ecosystems before ultimately ending up in your food, water, and air. The story begins with plastic production and use. As we've noted, global plastic production has increased exponentially from 2 million metric tons in 1950 to nearly 400 million metric tons in 2021. Most of this plastic is used for packaging, much of which is single-use, designed to be discarded after a brief period of use. Despite recycling efforts, less than 10% of all plastic ever produced has been recycled. The rest accumulates in landfills, is incinerated, or leaks into the environment. The ocean has become the ultimate repository for much of this plastic waste. An estimated 8 to 10 million metric tons of plastic enter the oceans annually. This plastic comes from multiple sources. Direct disposal at sea by ships and fishing vessels contributes, but the majority of ocean plastic originates from land-based sources. Plastic litter on beaches and riverbanks is washed into the ocean. Plastic waste in open dumps is blown or washed into waterways. Microplastic fibres and particles from washing clothes, tire wear, and industrial processes enter wastewater systems that discharge into rivers, which carry them to the sea. Once in the ocean, plastic begins to break down. Sunlight, particularly ultraviolet radiation, causes photodegradation, weakening the polymer chains. Wave action provides mechanical stress, fragmenting pieces into smaller particles. Over time, a plastic bottle becomes thousands of microplastic fragments, which continue breaking down into even smaller pieces. This process can take decades or centuries, but it's continuously ongoing with all the plastic in the ocean. Marine organisms interact with these microplastics in multiple ways. Filter feeders like mussels, oysters, and other bivalves filter large volumes of water to extract plankton for food. In the process, they also filter microplastics which accumulate in their tissues. When you eat shellfish, you're consuming whatever they've accumulated, including microplastics. Because shellfish are often eaten whole, including their digestive systems where microplastics concentrate, they're a significant source of human microplastic exposure. Fish also ingest microplastics, either directly by mistaking them for food or indirectly by eating prey that contain microplastics. Studies examining fish caught for human consumption have found microplastics in both the digestive tracts and the flesh of numerous species. While gut contents are typically removed during fish processing, microplastics that have translocated into the flesh remain in the portion you eat. But the food chain doesn't stop with individual organisms. Microplastics accumulate and potentially biomagnify as they move up food chains. A small fish ingests microplastics. A larger fish eats many small fish, accumulating their microplastic burden. A A top predator like tuna or swordfish eats many large fish. At each step, microplastics may accumulate, and since humans are often at the top of the food chain, we may be accumulating the concentrated burden from all the organisms below us. The ocean isn't the only aquatic environment contaminated with microplastics. Freshwater systems, rivers and lakes are also extensively polluted. Some studies have found microplastic concentrations in rivers comparable to, or even exceeding those in ocean surface waters. These freshwater microplastics come from wastewater discharges, agricultural runoff, industrial emissions, and atmospheric deposition. Drinking water, whether from surface sources like rivers and lakes or groundwater wells, contains microplastics. Multiple studies across the world have confirmed this. A comprehensive survey found microplastics in 83% of tap water samples from 14 countries on 5 continents. The United States had the highest contamination rate at 94%, but Europe and Asia also showed high rates. The concentration varies, but can reach several fibres or particles per litre. Wastewater treatment plants, while they remove many contaminants, are not effective at filtering out microplastics, especially the smallest particles. Some microplastics are captured in sewage sludge, which is often applied to agricultural fields as fertiliser, leading to soil contamination. But many particles pass through treatment and are discharged into receiving waters. The treated water that comes out of your tap has been through filtration processes, but these processes weren't designed to remove microplastic particles, especially nanoplastics. Bottled water, ironically, often contains more microplastics than tap water. The plastic bottles themselves shed particles into the water, especially when exposed to heat or stored for long periods. Studies have found that bottled water can contain anywhere from tens to hundreds of thousands of microplastic particles per litre, depending on the brand, bottle material and storage conditions. When you choose bottled water thinking you're avoiding contaminants, you may actually be increasing your microplastic exposure. Agricultural systems are another important pathway. Microplastics contaminate soils through multiple routes. Plastic mulch films used in agriculture break down, leaving microplastic residues in soil. Sewage sludge applied as fertilizer introduces microplastics. Atmospheric deposition adds particles that settle from the air. Irrigation with contaminated water brings microplastics to fields. Once in soil, microplastics can be taken up by plants. Studies have shown that crop plants including lettuce, wheat, carrots, and others can absorb microplastic particles through their roots. These particles can translocate to edible plant parts, meaning that fruits and vegetables may contain microplastics, even if they're grown far from industrial areas. Research is still determining how common and significant this pathway is, but it means that even plant-based foods may contribute to microplastic exposure. The atmosphere is also contaminated. Microplastic fibres and fragments are suspended in air, blown up from surfaces or released directly from sources like tileware. Studies measuring airborne microplastics have found them in both outdoor and indoor air, with indoor concentrations often higher due to sources like synthetic textiles and carpets. Indoor air in homes and offices can contain significant concentrations of microplastic fibres, primarily from synthetic textiles. Every time you sit on a couch, walk on carpet, or handle clothing made from synthetic fibres, you're releasing microfibres into the air. These fibres settle as dust and remain suspended in the air you breathe. Studies estimate that indoor air exposure contributes substantially to total microplastic intake, potentially rivaling or exceeding intake from food and water. The environmental journey of microplastics can span vast distances. Atmospheric currents can transport lightweight microplastic particles thousands of kilometres from their source. Microplastics have been found in remote pristine environments, including mountain lakes, Arctic ice, and deep ocean sediments. There is essentially nowhere on Earth that remains free from microplastic contamination. This global distribution has been referred to as the plastification of the planet. Just as carbon dioxide from fossil fuel combustion has increased in the atmosphere globally, Microplastic concentrations are increasing in all environmental compartments worldwide. And unlike carbon dioxide, which can be absorbed by natural processes, plastic persists indefinitely. Understanding these environmental pathways is crucial because it reveals that microplastic exposure is essentially unavoidable given current conditions. Unless you can avoid eating food, drinking water, and breathing air, you're being exposed to microplastics. The question isn't whether you're exposed, but how much and what the consequences are. To truly grasp the magnitude of microplastic contamination, we need to zoom out and look at the global scale of the problem. This isn't a localized issue affecting a particular region or population. It's a planetary phenomenon that has fundamentally altered the composition of Earth's ecosystems within the span of a single human lifetime. Consider the oceans. The Great Pacific Garbage Patch, that infamous accumulation of plastic debris, covers an area estimated at anywhere from 700,000 to more than 1.6 million square kilometres, depending on the criteria used to define it. That's roughly twice the size of Texas, and it's not the only such accumulation. There are similar, though smaller, plastic accumulation zones in other ocean basins. But these visible accumulations of floating plastic represent only a fraction of ocean plastic pollution. Most ocean plastic eventually sinks or breaks down into microplastics that become dispersed throughout the water column and accumulate in sediments. Studies have found microplastics from the ocean surface to the deepest ocean trenches, more than 10,000 metres below the surface. They've been found in sea ice in both the Arctic and Antarctic. Essentially every cubic metre of ocean water contains microplastic particles. Marine sediments are becoming increasingly contaminated with microplastics. A study examining sediment cores, layers of accumulated sediment that provide a historical record similar to tree rings, found that microplastic concentrations have been exponentially increasing since the 1940s, tracking the increase in plastic production. The sediment is essentially becoming fossilized with plastic, creating a permanent marker in Earth's geological record of the Anthropocene, this human-dominated epoch. Terrestrial environments are similarly contaminated. Studies examining soils across the world have consistently found microplastics, with agricultural soils often showing particularly high concentrations due to application of contaminated sewage sludge and use of plastic mulch films. Some agricultural soils contain more microplastic pollution by mass than ocean surface waters. Freshwater environments, which provide drinking water for much of the global population, show widespread contamination. The Great Lakes in North America contain an estimated tens of thousands of tons of microplastic. European rivers show similar contamination. Asian freshwater systems, particularly in densely populated regions, often show even higher levels. Atmospheric transport means that microplastics have reached even the most remote terrestrial environments. They've been found in pristine mountain lakes in the Alps and Pyrenees, in Arctic snow, in Antarctic surface waters. A study of remote US national parks found microplastic contamination in all sites sampled, including highly remote areas far from population centres. The plastic is literally falling from the sky as atmospheric deposition. The biodiversity impacts are profound. Over 300 species have been documented to interact with plastic, either by ingestion or entanglement. For marine organisms in particular, plastic ingestion is widespread. Studies have found plastic in the stomachs of sea turtles, seabirds, whales, fish, and invertebrates across all ocean basins. Some seabird species show plastic ingestion rates exceeding 90%, meaning virtually every individual bird has plastic in its digestive system. The ecological consequences extend beyond individual organisms. Microplastics can alter sediment properties, affecting organisms that live in or rely on sediments. They can interfere with nutrient cycling and primary productivity in aquatic systems. Attached biofilms can alter ocean chemistry and create novel habitats for microorganisms, potentially affecting marine microbial communities that play crucial roles in global carbon and nutrient cycles. The scale of human exposure follows from this environmental contamination. With microplastics ubiquitous in air, water, food, and consumer products, human exposure is essentially universal in developed countries and increasingly common in developing countries as plastic use increases. Biomonitoring studies looking for microplastics in human samples have consistently found high detection rates, whether examining blood, urine, stool, breast milk, or tissues. The temporal trend is particularly concerning. As we've seen with the brain tissue study comparing 2016 and 2024 samples, microplastic concentrations in human tissues appear to be increasing over time. This suggests that despite growing awareness of the plastic pollution problem, we're losing ground. Environmental concentrations continue to increase as legacy plastic breaks down, and new plastic is produced and eventually enters the environment. Projections for the future are sobering. If current trends continue, plastic production could increase to over 600 million metric tons annually by 2040. With waste management systems unable to capture all this plastic, environmental leakage would increase proportionally. Some models predict that the mass of plastic in the oceans could exceed the mass of fish by 2050. The economic costs are difficult to quantify but likely substantial. Damage to marine industries including fishing and tourism, costs of clean-up efforts, and potential healthcare costs related to human health effects of microplastic exposure could collectively reach into the billions of dollars annually. There's also an environmental justice dimension to this issue. While plastic pollution is global, the burden is not evenly distributed. Lower-income communities and countries often have less effective waste management systems, leading to higher environmental contamination. Communities of colour in the United States have been shown to have higher exposures to various environmental pollutants, and there's concern that microplastic exposure may follow similar patterns. Developing countries, particularly in Asia, bear a disproportionate burden of plastic waste, including imported plastic waste from developed countries. The intergenerational justice aspect is also profound. The decisions to mass-produce plastic and inadequately manage plastic waste were made primarily by previous generations, but the consequences will be borne by current and future generations. Children today are growing up in a world where they're exposed to microplastics from conception, and the full extent of what this means for their health won't be known for decades. This brings us to perhaps the most important question. Given the scale of the problem and the evidence of health effects, what can be done? What does the research tell us about how to address this planetary contamination and protect human health? After examining the extensive evidence of microplastic contamination and its potential health effects, you might expect that we have clear answers about the risks and clear guidance on what to do. Unfortunately, that's not quite the case. While research has advanced rapidly in recent years, particularly since 2020, there remain enormous gaps in our understanding. These gaps make it difficult to provide definitive answers about health risks and complicate efforts to develop effective mitigation strategies. Let's start with what we do know with reasonable confidence. We know microplastics are ubiquitous in the environment and in human bodies. This has been demonstrated conclusively across numerous studies. We know that humans are exposed through multiple pathways including ingestion, inhalation, and possibly dermal contact. We know exposure is continuous and likely increasing over time. We know that microplastics have been found in numerous human organs and tissues including blood, lungs, liver, kidneys, placenta, brain, testicles, and others. We know concentrations vary, but are substantial enough to be detected with current analytical methods. From animal studies and cell culture experiments, we know that microplastics can cause oxidative stress, inflammation, cellular damage, and alterations in gene expression. We know they can disrupt endocrine function through associated chemicals. We know they affect reproduction in animal models and alter development in offspring of exposed animals. From limited human studies, we know that microplastics in arterial plaque are associated with increased risk of cardiovascular events. We know that people with inflammatory bowel disease have higher microplastic concentrations in intestinal tissue. We know that microplastic levels in human brains are increasing over time. But there's a vast amount we don't know. We don't know the precise health risks associated with current levels of human exposure. We don't know whether there are thresholds below which effects don't occur, or whether any exposure poses risks. We don't know which types of plastic or which particle sizes are most harmful. We don't know whether the primary health threat comes from the plastic particles themselves or from the associated chemicals. We don't know the long-term consequences of lifelong exposure beginning in the womb. We don't know whether microplastics are contributing to observed trends in declining fertility, increasing rates of cancer, rising prevalence of neurodevelopmental disorders, or other population health changes. We don't know how much microplastic exposure is coming from different sources for a typical person. Estimates vary widely, and exposure likely differs substantially between individuals based on their diet, lifestyle, and environment. We don't know how long microplastics remain in the body, or whether they accumulate over time to reach ever higher concentrations. Part of the challenge in answering these questions is methodological. Detecting and quantifying microplastics in environmental and biological samples is technically difficult. Different studies use different methods, making it hard to compare results. Many studies can only detect particles above a certain size, missing the nanoplastics that may be most numerous and most able to penetrate tissues. Standardization of methods is improving, but we're still in relatively early stages. There's a need for better analytical techniques that can detect smaller particles, identify plastic types, and quantify concentrations more accurately. There's also a need for biomarkers, measurable indicators of microplastic exposure and effects that could be used in epidemiological studies to track exposure and health outcomes across large populations. Another challenge is establishing causation. Many of the human studies showing associations between microplastics and health outcomes are observational, meaning they can demonstrate correlation but not prove that microplastics cause the health effects. It's possible that microplastics are markers of other exposures or lifestyle factors that are the actual causes. Conducting randomized controlled trials, the gold standard for establishing causation, is not ethical for potentially harmful exposures. You can't deliberately expose people to high levels of microplastics to see if they develop health problems. Instead, researchers must rely on observational studies, animal experiments, and cell culture work to build evidence. The time lag between exposure and health effects also complicates research. If microplastic exposure during childhood affects cancer risk in adulthood, you'd need to follow people for decades to detect the effect. Such long-term studies are expensive and difficult to conduct. Despite these challenges, research is advancing rapidly. The field of microplastics research has exploded in the past 5 years. Major research initiatives have been launched, including the Momentum project in Europe, which aims to integrate research efforts to understand health effects of micro and nanoplastics and develop solutions. Funding for microplastics research has increased substantially as recognition of the problem has grown. Some emerging research directions are particularly promising. Advanced imaging techniques are being developed to track microplastics in living organisms at high resolution, which could help understand how they move through the body and where they accumulate. Mechanistic studies are working to understand exactly how microplastics cause cellular damage and what molecular pathways are involved. This could identify potential therapeutic targets or biomarkers. Epidemiological studies are being launched to examine associations between microplastic exposure and various health outcomes in large human populations. Some of these studies are following pregnant women and their children to examine effects of early life exposure. Others are looking at occupational exposures in workers who handle plastics to see if higher exposures are associated with increased health problems. There's also growing research on interventions to reduce exposure. Studies are examining which dietary changes or lifestyle modifications can meaningfully reduce microplastic intake. Research on air filtration, water treatment, and other technological solutions is advancing. Policy-relevant research is examining which regulatory approaches might be most effective at reducing environmental microplastic levels. From a public health perspective, The precautionary principle suggests that we shouldn't wait for complete certainty about risks before taking action, especially when dealing with exposures that are widespread, increasing, and potentially harmful. Even if we don't know the exact magnitude of health risks, the evidence suggesting potential harm, combined with the feasibility of reducing exposure through various means, argues for taking steps to minimize exposure and reduce environmental contamination. On an individual level, there are practical steps you can take to reduce microplastic exposure, though it's important to recognize that individual actions alone cannot solve a systemic problem. These include choosing glass, stainless steel, or ceramic containers for food and drink storage instead of plastic. Avoiding microwaving food in plastic containers as heat increases leaching of chemicals and particles. Choosing natural fiber clothing and textiles when possible, as synthetic fabrics shed microfibers. Drinking tap water filtered through a quality filter rather than bottled water. Eating fresh, unpackaged foods when feasible. Avoiding cosmetics and personal care products that contain microplastics or plastic ingredients. Vacuuming and dusting regularly to reduce indoor dust that contains microfibers. At a broader level, addressing the microplastics crisis requires systemic changes. This includes reducing plastic production overall, particularly of single-use plastics that quickly become waste, improving waste management systems to prevent plastic leakage into the environment, developing truly biodegradable alternatives to conventional plastics for applications where alternatives are feasible, investing in technology to capture microplastics from wastewater before discharge, requiring manufacturers to disclose chemicals used in plastics and phasing out the most harmful additives, supporting cleanup of existing plastic pollution in oceans and other environments. Some regulatory progress has been made. Many jurisdictions have banned plastic microbeads in personal care products. Some have restricted single-use plastic bags or food containers. The European Union has been particularly active in regulating plastics, including banning certain plastic products and requiring member states to collect and recycle specific percentages of plastic waste. But globally, plastic production continues to increase, and waste management systems remain inadequate. The economics of plastic production favour continued growth, as plastic is cheap to produce, especially when oil prices are low, and demand remains high across all sectors of the economy. Without concerted international action, the trajectory toward ever-increasing plastic production and environmental contamination will continue. The United Nations Environment Programme has been working toward a global plastics treaty, an internationally binding agreement to address plastic pollution across the life cycle, from production to disposal. Such an agreement could set global targets for reducing plastic production and waste, establish common standards, and coordinate international action. But negotiations are complex and success is not guaranteed. There's also the question of remediation. Even if we completely stopped producing plastic today, the legacy of past production will persist in the environment for centuries. Existing plastic will continue breaking down into microplastics for generations. Some researchers are working on technologies to capture or remove microplastics from the environment, but these face enormous technical challenges given the scale of contamination and the small size of particles. Looking forward, we face critical decisions about the future of plastics in society. Do we fundamentally rethink our relationship with plastic, moving away from the disposable culture that has developed over the past 70 years? Do we invest in developing and scaling up true alternatives that don't persist in the environment? Do we accept that some level of microplastic contamination is now inevitable and focus on managing the most harmful exposures? These are not just technical or scientific questions. They're social, economic, and ethical questions about what kind of world we want to inhabit and what legacy we want to leave for future generations. The dark truth about microplastics inside your body is that we're all participants in an uncontrolled global experiment. We're the first generations to live in a world saturated with synthetic polymers, and we're discovering in real time what happens when these materials infiltrate our bodies. The full consequences won't be known for decades. but we know enough to recognise that this is a problem that demands urgent attention and action. The particles inside you are small, but their implications are enormous. They represent a fundamental alteration of what it means to be human in the 21st century. We are no longer biological beings inhabiting a natural world. We are hybrid creatures, combinations of biology and synthetic materials, living in environments that are equally hybrid, part natural and part artificial. Whether this transformation proves to be harmful in ways that fundamentally affect human health and well-being, or whether it turns out that our bodies can coexist with low levels of microplastic contamination without major consequences, remains to be seen. But one thing is certain: the presence of plastic fragments in every organ of your body represents an unprecedented change in human biology, one that we're only beginning to understand. The question is not whether microplastics are inside you. They are. The question is what we're going to do about it. The research in this area is evolving rapidly, and I'll continue to cover new developments as our understanding grows. Let me know in the comments what aspect of microplastics you're most concerned about, or what topics you'd like me to cover next. Thanks for watching, and take care out there in our increasingly plastic world.